Well, hello everybody, and thank you for the nice introduction, and thank you for giving me the opportunity to present our data here at this Congress. And yeah, so basically our main goal is to develop an in vitro model of the lung to establish respiratory viral infection on this model, and in particular we were interested in SARS-CoV-2 infection here. So let me just quickly tell you what is our motivation to develop such a model. So first of all, we all know that the major target of SARS-CoV-2 is the distal lung, where we have most severe manifestation of the disease. The major target cell type in the distal lung are actually the type 2 alveolar epithelial cells, which are very important to maintain a healthy alveolar epithelium and enable breathing, basically the breathing of the alveoli, that they maintain their function. In the most critical phase of the disease, usually this manifests as diffuse alveolar damage. You can have microvasculature activation, and also in the most severe case, then the breakdown of the air-blood barrier, which can then lead to the death of the patient. And thanks to several measures that have been undertaken, the pandemic has been turned into an endemic viral disease by now. And besides monitoring and vaccination, therapeutic development still remains an important aspect actually in controlling the disease. And I hope Rebecca has already convinced you that it's still a big challenge to actually develop novel therapeutics and also find the ones that are then really working in the patients after having done all the drug screens. But still it's an important aspect or an important part to protect, for example, immune deficient patients from disease which cannot be vaccinated. It's also important to prevent severe disease or to accelerate recovery. In the end, it could also be a cheap alternative for vaccination in countries that don't have easy access to vaccination. And of course, it will also help to increase our preparedness for future pandemics if we have more specific therapeutics for coronaviruses. And yeah, as Rebecca already mentioned, the major challenges in drug development for the respiratory infectious virus diseases are actually the lack of relevant models that we have. So for the high throughput screening models, it's a major problem that we don't have really relevant cell models available. So often there are non-human cell lines used or cell lines that are not in the appropriate maturation state. And also there are rather simplistic models, and therefore it's really hard to translate the findings from these in vitro screens then to the clinic, because they just lack in vivo complexity. But also the in vivo models have some limitations, mainly due to interspecies differences. So in the immune response, for example, also in the breathing rate and in the breathing pattern of animals and humans, in lung anatomy, and also in the host cell tropism of the virus itself. And so this can lead, for example, to discrepancies in the manifestation of the disease or in the severity of the disease, which is then also a challenge. And in this project, actually, we aim to address this lack of predictive human-derived in vitro models and close the gap between the high screening in vitro models and the animal models. And how are we planning to do this? So basically we aimed now to develop a drug screening pipeline for the distal lung, so for the alveoli, by combining two different model approaches. So on one hand, we would develop a medium to high throughput model of the alveoli on transwell inserts, and on the other hand, we would derive a more physiological lung-on-chip model that would allow us to study or hopefully better predict responses to therapy, and then only select the most promising compounds for further development. And as I already mentioned, so we have now focused on the optimization of this platform for SARS-CoV-2 infection, but of course we also hope that we can apply this platform then for other respiratory virus infections. So as I already mentioned, the major target of this virus is the alveolar epithelium. So basically we started selecting primary alveolar epithelial cells or optimizing infection for these cells and just see if we can infect them on the transwell inserts model. So what we did, we isolated alveolar epithelial cells from donors and seeded the cells on the transwell inserts. We matured them in a liquid interface and then infected them with the virus. And you can see that we also have cells that express AC2 and TMPRSS2. So these are two proteins that are very crucial for viral entry into the cells. So basically it's a prerequisite that these proteins are expressed in the cultures to actually be infected. And we found that the virus infected these cultures of this donor 1 and also increased over time. So we see more cells infected at 72 hours post-infection than at 48 hours post-infection. And this is actually also the same when we determine the virus titer for this donor 1. So it actually creates or produces quite high viral titers at 72 hours post-infection. We really see a productive infection in this patient. So we were very happy, but then we tested 3 other donors, and there we were less lucky apparently. So we had 2 donors that seemed to be resistant to the virus, so we didn't get any infection in these 2 donors, and 1 donor that was infected by the virus, but it just produced very low viral titers. So this led us to the conclusion that the primary cells are actually very nice because they might capture some intrinsic differences in the donor-to-donor variability, how different people react to the infection actually with SARS-CoV-2. So it would be very interesting also to check if we find some underlying intrinsic factors that actually lead to these differences that we see in our in vitro model. However, for our purpose, it was not ideal because we want to develop a robust and reproducible drug screening platform. And that's why we decided we switched to a cell line. And the cell line that we chose is actually is the AX immortalized alveolar epithelial cells that we got provided from Alveolix. So this cell line is derived from primary alveolar epithelial cells. It's human-derived, and it contains a mix of type 1 and type 2 alveolar epithelial cells. And you can see this cell also forms a tight barrier on the transwell inserts, and it's stable over long-term culture. And of course, for me, it was important to know if it could also be infected by the virus. So I checked for the expression of AC2 and TMPRSS2 in this cell line, and you can see it's the protein, both proteins actually are expressed in these cells. And also we saw an increase of expression of gene expression for AC2 and TMPRSS2 when the cells were matured in a liquid interface. So this is quite interesting, and we were also wondering then if the infection would be more efficient in a liquid interface or not, and that's actually what we tested next. So we used these cells, we seeded them on the transwell inserts, and on one hand, we either matured them in a liquid interface for at least 8 days before infection, or we kept them in submerged culture. And you can see in the immunofluorescence images actually that in both cases we have cells that are infected, but there are really strikingly more cells infected when the cells were matured in a liquid interface before infection. And you can also see that in the viral titer that is reached, and it's really significantly higher in cultures that were exposed to air from the apical site before we infect them and have been matured. Besides the virus, we were also evaluating the, let's say, the TR or barrier integrity of the epithelium, basically as a measure for the functionality and the health of the barrier, of the epithelial barrier. So we measured TR in both cases, and as you can see in the left graph of the TR measurements, it's a submerged condition, and there you can really see, despite the infection with the virus, after 48 hours we still have a very high TR value, and the cells do not respond at all to the presence of the virus, so they're just growing happily. And only when we have the cells in a liquid interface, so on the graph on the right, I hope you can see it behind me, we see actually at 48 hours the breakdown of the alveolar barrier, so the cells are being killed and they really lose their ability to form a tight barrier. So this was actually quite nice. So we had our model, so we really have to choose the liquid interface model, and we further developed this then for drug screening. So that was the next thing that we tried was actually to develop a drug screening protocol. At first we started testing remdesivir just as a positive control because it is standard of care in patients, and also it has been well documented in the literature that it works very nicely also in vitro against SARS-CoV-2 virus. And what we do, it's pretty similar than what I was telling you before, the protocol. So we use our liquid interface model, and then at day 14 we infect the cells from the apical side, and we also start treatment at the same time when we add the virus from the apical and from the basal side. However, it's very important that we remove again the inoculum from the apical side after 2 hours, and we wash off the virus and re-establish a liquid interface just to maintain the correct maturation of the cell for infection. So then we have for 48 hours the drug treatment just from the basolateral side. And you can see, I hope you can appreciate from the bright field images that once the virus is present, we have a cytopathic effect visible already in our transwell cultures. And as soon as we treat with remdesivir, we see that with increasing doses, remdesivir is really able to prevent the cytopathic effect. And basically we have again an epithelium that looks quite healthy. So we measured the TR value again as a readout for the functionality of the barrier. And you can see also here, once we add the virus and don't treat with a drug, we have a significant decrease in the TR. However, once we add remdesivir, we see a dose-dependent response to the treatment, and basically it is able to reconstitute barrier function at a nanomolar level. So it is really efficient, and it validated our model that it really seems to work. And this encouraged us to test 2 experimental compounds for which it's not so clear how they function. So on one hand, we tested cyclosporine, which from in vitro screens, it has been shown that it has some antiviral effect. However, in clinical trials, the results are still controversial. And in our model, actually, when we treated with cyclosporine, we saw a little increase in the barrier function, but it is really not reconstituted to uninfected control. So in our model, cyclosporine is really not efficient. And the second compound that we tested was amiodarone, which is also similarly as cyclosporine, seems to be not really or not significantly improving the disease outcome in COVID-19 patients. And in our model, we see a trend that once we add amiodarone, at least in the highest concentration, the barrier function is a little bit improved, but still it's not at the same level as the untreated control, and it seems to not work very nicely. So this gives us this drug screening platform at hand for high throughput screening. However, it has to be considered that we are here looking only at the one cell type, and also we were not able to really have the complete breakdown of the barrier. So I don't know if you noticed, but even in the presence of the virus, the TR remains quite high, so there is still a barrier present. So we don't have this complete loss of or complete lung damage and the breakdown of the air-blood barrier in this model system. And that's why we also developed a lung-on-chip model to also study the damage in a more physiological setting. And basically, the lung-on-chip models are actually aiming to just recapitulate this very thin air-blood barrier that you have in the alveoli and the low gas exchange. And it's created basically or mainly from the endothelial cells and from the type 1 and type 2 alveolar epithelial cells that form this very thin barrier. And of course, you also have some immune cells in the alveoli that protect you from infection from inhaled pathogens. However, once the lung is infected with SARS-CoV-2, this can lead to severe manifestation and to COVID-19 pneumonia. So you can have a massive inflammation in the alveoli, which is associated also with cell death, with syncytial cells, with type 2 alveolar epithelial cell hyperplasia, and microvasculature activation. And this can then lead to lung damage. So what we chose as our chip model was the AX12 chip from Alveolix. So this chip comes in a 96 well plate format, and you can see that we see the cells. So one unit is basically composed of 3 cells, of three wells, and we only see the cells in the central well. In our model, we chose again the alveolar epithelial cell line that we used before, also on the transwell inserts, and the human lung microvasculature endothelial cell for the representation of the endothelium. And you can see they form a barrier in these chips. And we also checked for the presence of some crucial factors in our culture system. So we have a good proportion of type 2 alveolar epithelial cells, which are the major target of the virus, and also they express AC2 and TMPRSS2, which are very important host factors that allow the virus to enter. And actually one advantage of the Alveolix chip is that we can implement a stretch. And I would just like to briefly explain you how this actually works in this chip. So just focus on the cell culture well, so the central well, where we also culture our cells on the membrane, which is depicted in blue. And what Alveolix was actually doing, so they have the motion inspired by the breathing motion, so by the movement of the diaphragm, and what they did is just introducing microdiaphragm at the bottom of the plate, which you can see in gray, and this can be deflected by application of negative pressure, and this deflection is then transmitted to the cell culture membrane, again in blue, and this allows then this 3D stretch, as you can appreciate in the movie, so pulling down the membrane, and simulating stretch in a physiological pace and manner. So of course we are interested also in how stretch would affect the infection or the outcome of COVID-19. So we compared static condition to dynamic condition in our lung-on-chip model. Again, we have the model with the alveolar epithelial cell seeded on the apical site in a liquid interface matured. And shortly after establishing a liquid interface, we also implement stretch. And then we infect the co-culture with SARS-CoV-2 at day 16. And you can see in dynamic culture, the barrier is a bit lower, so the TR values are a bit lower, actually in a more physiological range than in static condition, but it remains stable. And probably this is also a hint that we have a bit of a higher proportion of type 2 alveolar epithelial cells. So once I add the virus on this model system, you can see actually also in static and also in dynamic we have infected cells. So the virus is able to infect our alveolar model here. However, for quantifying the virus titer from these two conditions, I actually saw that the virus titer is a little bit increased in the allostatic condition. So in implying that actually stretch somehow seems to enhance virus production a little bit in our model. And besides checking for the virus, I was also checking for lung damage in this model system, and actually there I saw a striking difference between static and dynamic conditions. So you can see here just the bright field images, again from the same experiment, with the static condition on top and with the stretch applied on the bottom row. So you see on the left panel there is just a culture right before infection, so we have a nice monolayer there. And 48 hours post-infection on the right panel, you can see that in static conditions on the top right, there are some patches of syncytial cells or hyperplastic cells visible, but not a lot. Whereas when we add stretch, I think you can appreciate that there is much more damage induced to the epithelium. And this can also be seen once we check for the barrier integrity. So what we saw is actually that in static condition, the TR decreases significantly, but still it remains at a very high level, and we still have a functional barrier. And in dynamic condition, there it's really, we have a 90% reduction of the barrier, of the TR, and it's really a complete breakdown of the barrier. So this basically also highlights the importance of stretch to mimic lung damage. And finally, we have also used this model to screen for drug. We have again tested remdesivir. It's a very similar protocol as we used on the transval insert. So we start the treatment when adding the virus, and then re-establish a liquid interface and just do the treatment from the basal side. And also here you can see when the virus is present but untreated, we already have some cytopathic effect. However, in the presence of remdesivir, it seems to protect the barrier from virus-induced damage. And this you can also see by the TR measurement in the graphs. So when the virus is present, we have the breakdown of the barrier, and remdesivir maintains an intact barrier at 48 hours post-infection. So with this, I would like to conclude. So we have developed this Alvirax platform for drug screening purposes for SARS-CoV-2 medications. On one hand, on the transval inserts, here we have actually reached a high virus production, which is very efficient, and also we have demonstrated the importance of a liquid interface to infect the cells. And we have the lung-on-chip system, which is mainly important to study the physiological stretch and to model the lung damage that is induced by the virus. So we hope actually that with this platform we manage to better predict candidate compounds, so to reduce the costs and the time until a drug gets finally into the market, but also to reduce animal testing, and it also allows to work with human-derived cells and could also be used for personalized healthcare. And just as a very brief outlook, of course, we were also wondering if this could also be applied for other respiratory viruses. So we had just very recently started a pilot experiment with influenza A virus on the transval inserts, and you can see also influenza A virus is able to infect our cultures and produce actually a lot or high viral titers in our system. And finally, it could also be applied to study host pathogen interaction in basic research, as we have also demonstrated the importance of microenvironment cues like a liquid interface and stretch. And with this, I would like to thank everybody involved in this project, so my group from University of Bern, our collaborators, and our implementation partner. And thank you all for your attention.